In this lesson, we'll continue our discussion of enzymes. What we'll do is we'll take a look at, in more detail and more specificity, enzyme catalyzed reactions, what's happening. And then in our first lesson on enzymes, we introduced the concept of cofactors and coenzymes. Let's look at those in a little bit more detail also. So we can think of enzyme catalyzed reactions occurring in two stages. The first stage is the formation of something known as the enzyme substrate complex. You may hear me refer to that as the ES complex. Just so you know, you may hear that. The second step is going to be the conversion of the substrate into the product, and then the release of the enzyme kind of occurring simultaneously. The second step is the slower step that limits the rate of the reaction. So I left you some room to draw that little space there. Again, I wasn't being lazy. I just didn't know I would be able to fit it all on one line. So E stands for enzyme. If we add that to the S, which is the substrate, we form this ES complex, right? So ES is just the ES complex, right? So what we did basically, we're going to see is combine the enzyme and the substrate. Now the chemical reaction is going to begin to take place. We're going to enter something known as the transition state, the ES transition state. That eventually is going to form the product, but the enzyme is still attached to the product. So that, guys, you're going to call the EP complex. And then finally, the product is going to separate from the enzyme. And if you remember from our first lesson, the enzyme is always unchanged. So if you want to draw that down, we'll take a look at what's happening first in this ES complex in a little bit more detail. All right, so basically, again, just as a review, we take the enzyme, we combine it to the substrate, we form the ES complex. Once they're formed, the chemical reaction starts to take place and we get the transition state. Eventually the product is going to be formed, but the product is still attached to the enzyme. So this we're going to call the EP complex, and then eventually the product and the enzyme are going to separate. Again, the enzyme is always going to be unchanged. So what we're looking at first is this ES complex. So let's take a little bit of a detailed look at the active site on the enzyme. What is it, right? It is nothing more than a pocket or a cleft on the surface of the enzyme. The shape of the active site complements the shape of the substrate. If you remember from our first lesson, we said that the enzymes have specificity, right? So it's like your key going into your car door, as I mentioned in the previous lesson, right? Your key has to fit your car door. Now, the enzyme will hold the substrate by hydrogen bonds, van der Waals forces, electrostatic attraction. So we see these interactions that we studied in our very first modules. This is how the substrate and the enzyme stay attached. Now, the conformation of the active site determines the specificity of the enzyme. As we said earlier, only a substrate that fits is going to be able to enter the chemical reaction. If a substrate doesn't fit the enzyme, right, that enzyme has to find another substrate. So there's two theories on this. The original theory of how the enzyme will connect to the substrate was called the lock and key model, right? This is the original theory where the enzyme and the substrate actually snap together. So I'm going to jump ahead for a second. This is a picture from our book where they show you the enzyme. This area here is the active site, and then here's the substrate. If you notice, right, these areas of the substrate will fit beautifully right into the active site, and this green substrate will snap into place, and now we have our ES complex. Right, that was the original thought, known as the lock and key model. The newer theory is now known as the induced fit model, where we see that the substrate and the active site are not exact. They're not an exact fit. What will happen is this. I'll come back to the note. Let's go to the second picture. This is also from our book. Notice that the active site kind of resembles the substrate area here, but it's not a perfect fit. What happens is once the substrate comes up to the active site, the enzyme will actually conform to the substrate to make the perfect fit. So they call this the induced fit model, right? This is the newer theory. The active site of the enzyme is considered to be flexible and will approximate the shape of the substrate. Right? The active site will mold itself around the substrate to produce that perfect fit. So that's what we think is happening in this newer theory. All right, so again, this is the old lock and key model, and this one we're going to call the induced fit model. Again, close, right? But doesn't fit exactly. But once they come together, the enzyme will conform around the substrate to get our ES complex. Okay, so again, still talking about the enzyme substrate complex, just to go a little bit more into this enzyme specificity. So again, specificity is nothing more than the ability of the enzyme to bind to only one or maybe a few substrates, right, that are very similar in structure. The enzymes are highly specific in the reactions that they catalyze. We had already gone over that, and their choice of reactants. For example, there's a whole types of category of enzymes known as proteolytic enzymes that help to break peptide bonds, right? So these proteolytic enzymes are only going to go into these types of chemical reactions. They're not going to go into reactions that break down sugars, that type of thing. So here's a picture from our book. Hopefully you recognize this as a protein. Remember the backbone? I always said look for NCC, NCC, and so forth. So this is our protein backbone, and then we have the R group sticking off the sides. Here's our amino group over here. That type of thing. So this would be the peptide bond right here. So a proteolytic enzyme will come in and break this bond so that we now have one protein over one, I should say one amino acid over here and then an amino acid over here. Right? That proteolytic enzyme only worked at that spot. It only worked at that place where we are going to break the peptide bond. Okay, so those are the main things that we want to know. Just go back to here about the ES complex. Let's take a look now as we move into the transition state. Right, the transition state. So once the ES complex is formed, the substrate becomes no longer energetically stable. Okay, this is known as the transition state. The substrate is in a state of being kind of like an in-between form. I put here an intermediate form where it actually has features of both the substrate as well as the product, right? So think of it again. What does transition mean? We're in a state of change, right? We're in a state of change. So things are changing now. So during the transition state, the following help to speed up the chemical reactions. The enzyme can help to break down bonds. It can help to also orient the substrates, bringing them closer together, right? So it actually changes the position of the substrates, right, so that they are nice and close together to make our product. And the active site of the enzyme may also change the pH of the area surrounding the substrate. So again, if we have like an optimal pH, that is also going to help with the chemical reaction to go forward. Right? So this transition state has a bunch of different things that are happening. Again, the enzyme helps to maybe break bonds if necessary, help to position the substrates, and then also even change the pH in the area, right, to help get an optimal pH. Okay, so from there, once the transition state has entered and we now start to form the product, we now get the EP complex, right? If you go back to your, I don't want to flip back again, but if you go back to your original diagram there, we're going to be at the EP complex. So this follows the transition state. At this point, the product is formed, but it is still attached to the enzyme. Okay, so that we're going to call the EP complex. And then finally, the product is going to be released from the enzyme, and then the enzyme is going to remain unchanged and can go into the same type of chemical reaction again. Right, and that's my last thing here. That's the release of the product. Right, the product separates from the enzyme, and now the enzyme is going to be released. It's unchanged, and it can do it over and over again. Okay, so the last thing I want to take a look at is we had mentioned in our first lesson that many enzymes require cofactors. So let's take a look at what some of these things are so that we have an idea. So that's my first line right here. Many enzymes require an additional non-protein group to function. We had already done these definitions. I just thought I'd throw them back in there again. Please know that an enzyme that is not bound to a cofactor we call an apoenzyme, whereas an enzyme that is bound to the cofactor is known as a holoenzyme. If you're following along on my lecture outline, we're going to see, guys, if you go to letter D under cofactors, cofactors are classified, if you just want to highlight, as inorganic substances, and then secondly as coenzymes. All right, so we're going to show you there's two types or classifications of cofactors: the inorganic substances, right, which is what I have over here. These are things like zinc, magnesium, nickel, selenium, manganese, even potassium. Right? These are typically things that are metals and minerals, that type of thing. Where coenzymes are all organic molecules. Right? These are things that are more organic. Now, we're going to break this down into two types of coenzymes. If you just want to highlight, the first ones are prosthetic groups. That's going to be number one on your lecture outline. And then if you go down a little bit, co-substrates. All right, so these are two different types of coenzymes. A prosthetic group is a coenzyme that is tightly bound to the enzyme. All right, so these are attached to the enzyme by covalent bonds, and they remain bound to the enzyme during the chemical reaction. Once the chemical reaction is over, that coenzyme is going to be released unchanged, and it can do it over and over again. All right, so this is what we would call a prosthetic group. Whereas a co-substrate, highlight right here, are loosely connected coenzymes. So they're bound, but they're more loosely connected, right? So they're going to be, they bind to the substrate via weak bonds, right? Not covalent bonds. But these are actually more involved with the chemical reaction. They can actually donate groups to the substrate. They can accept groups removed from the substrate, right? So they're kind of more involved in the chemical reaction. So since this co-substrate is involved with the chemical reaction and can be altered, we're going to find that other chemical reactions are going to be needed to convert the co-substrate back to its original form. And this is actually going to require energy. If we go back a slide, that prosthetic group, again, it just stays attached to the coenzyme. It's there during the chemical reaction. It's not changed. It's not altered at all. And all that's going to be is released once the chemical reaction is over. But the co-substrate, like I said, is going to be involved with the chemical reaction, so it's going to be altered. So we need to convert that co-substrate back into its original form, right? And that's what we're going to find would require energy. Okay, but those are both examples of coenzymes. So, like I said before, coenzymes are typically organic substances. So we typically find that coenzymes are vitamins, as an example. We're going to get into another lecture later in the semester as far as what a vitamin is. So just as an introduction today, think of a vitamin as an organic molecule. It's required in a diet in small amounts. Of all the water-soluble vitamins, which are the B and the C vitamins, only vitamin C is not associated with the known coenzyme, right? But we'll see that the B vitamins are, and we're going to look at them in more detail later in the semester. So just as some examples, vitamin B1, which is thymine, it forms a coenzyme known as thymine pyrophosphate. We usually abbreviate that TPP, and we're going to see that's involved in decarboxylation reactions where we're removing carbons. Vitamin B2 or riboflavin is a really important one. We're going to see this form something known as FAD, flavin adenine dinucleotide, which is important in carrying hydrogen atoms. Niacin, vitamin B3, same type of thing, also helps to carry negative hydrogen atoms known as hydride ions. These are both riboflavin and niacin. They are important in our chemical reactions that we're going to see later in the semester to produce ATP. Pyridoxine, vitamin B6, helps to carry amino and carboxyl groups. Pantothenic acid is going to be important for coenzyme A, which carries acyl groups. Biotin, which helps in carbon dioxide fixation. You don't have to memorize these now. I just wanted to show you this slide here, guys, and the previous slide. Don't really worry about these right now as far as memorizing them, as far as what they do. We're gonna see them later in the semester. I just want to impress upon you some of these important coenzymes and how these coenzymes are derived from vitamin B. Scorbic acid, though, which is vitamin C, doesn't have a known coenzyme. However, it does function in the hydroxylation of proline and lysine in collagen, right? We had actually talked about how important proline was in collagen. Vitamin C is actually important in that. But as far as a coenzyme, which is the topic of today, we don't really there's no known coenzyme. All right, so again, don't memorize these in the last two slides, this one and the previous one. Just kind of recognize the fact that B vitamins are important to us in forming coenzymes, and I have more for that, more for you later in the semester. What's our last slide here? So the last slide for this lesson, again, it's just what we just kind of went over. This is from our textbook, kind of summarizing some of these cofactors and coenzymes.